This video will be about Chapter 3 on amino acids, peptides, and proteins. Alright, this is your standard amino acid structure. You have 5 parts to it. You have your central carbon, you have your amine group, your carboxylic acid, a hydrogen, and an R group. Okay, amino acids are chiral, which means they are they have enantiomers of each other. Note that this is true for all the amino acids except glycine. And so there's an L and an R form of each amino acid, and L means that the amine group is on the left. And in biochemistry, we will most often see L forms of amino acids, and that's because the amino acids in the proteins in our body are mostly L. And just for a mnemonic, think L is for life. Okay, in life you see L. Alright, here's a big table of all of the amino acids right down here. Then you have the 3-letter abbreviation and the 1-letter abbreviation right next to that. And then you have the pKr over here. And you'll want to memorize the names of the amino acids, the 3-letter abbreviations, and the 1-letter abbreviation, and then these the 7 pKrs right here. And if you memorize these, then they're going to be really easy points on the test. And it just takes a little bit of time to memorize them, but they're guaranteed points if you do memorize them. And let's see. Okay, so with pKr, there's 7 amino acids, and the pKr means that the R group can develop a charge. There's 7 of them. With the non-ionizable R group, so the other 13, the R group does not develop a charge. And so if we go back to this picture, this amino acid, for the pKr, the amino acids with a pKr, this R group would develop a charge. Alright, so pKr, just a couple mnemonics. Tyrosine looks like tire, and so it's 10.07, so think I have 10 tires, and that's misspelled, of course, but it's just to help you remember tyrosine. That's actually how you spell tire in England. Okay, arginine, the 1-letter abbreviation is R, and so for arginine I just think of a pirate. You know, in pirates they're always saying R, and so think arginine. And so that will help you remember that this is R. And then the pKr is 12.5, and just think the movie Pirates of the Caribbean was so bad that I had to walk out after 12.5 minutes. And then let's see, for the 1-letter abbreviations, aspartic acid, glutamate is E, asparagine is N, glutamine, just think cutamine, and then lysine, think I like lysine. And so the K is the abbreviation for lysine. There's 5 groups of amino acids, and we're about to go through those 5. The first are non-polar, and note that glycine is the most simple with just a hydrogen. And remember, glycine is the only amino acid that is not chiral. Okay, alanine is just a methyl, and I just think of a guy named Alan. I don't know why that helps me remember it, but Alan has a methyl group. Proline is circular, and then valine, it looks like a V. The R group, see this V right here? So V for valine. And then leucine, you just add a methyl right in there, a CH2. And so you've got the V, but then you add a CH2. And then isoleucine should be like leucine, but just moved around. And so you just take this methyl group and you move it to the first carbon. Alright, for aromatic, you have phenylalanine. And so this pH sounds like an F, and so the F is the one letter abbreviation. Tyrosine looks like a tire, and it says tire right here. And so I think of tire for that aromatic with an OH coming off of it. And the Y in tire tells you that it's Y for the one letter abbreviation. Tryptophan looks weird, and so I just remember weird. W is the one letter abbreviation. Okay, next is polar. You've got serine. And then threonine looks just like serine. It just has one more methyl added onto it. And then cysteine is going to be an important amino acid because this is the amino acid that is used to make disulfide bonds with this sulfhydryl group. And so you take two cysteines and cram them together and you make a disulfide bond. And then remember, glutamine is Q for cutamine. Alright, so you've got the positively charged R groups, also sometimes called basic amino acids. And so my mnemonic for this is basically the history of Argentina is a lie. And apologize to anybody from Argentina, but it's just a, mnemonic. So basically the history, histidine of Argentina, arginine is a lie, lysine. And the pKr for histidine is 6, and remember arginine 12.5. Negatively charged amino acids. For aspartate, the abbreviation is D, and for glutamate, the abbreviation is E. This is showing that aromatic amino acids have an absorbance at 280 nanometers. And so if you wanted to decide what kind of amino acids were in a protein, you could run an assay through a spectrophotometer, which measures absorbance. And if you had some peaks at 280, you would know you had some of these aromatic amino acids. Alright, we're going to look at disulfide bonds now, and we have one cysteine right here, and we have a second cysteine right here, and if we cram those together, we get this disulfide bond right here. And so you'll want to know to make a disulfide bond, do you get oxidized or do you get reduced? Okay, and so I have another mnemonic up here, oil rig, and so the oil means oxidation is loss of electrons, reduction is gain of electrons. And so look right here, when we make a disulfide bond, we're losing electrons, and so that should be oxidation. So if you're asked on the test a question about disulfide bonds, you'll know that you need to oxidize it to make it, and if you want to break it, you need to reduce it. Okay, so there's some non-standard amino acids. You don't really need to know anything about the structure or the name, just that they exist. There's a lot of them. Here's two more, ornithine and citrulline. You'll see these later on in a couple months in the urea cycle. Okay, we're going to look at this thing called a Zwitter ion right here. Zwitter ion. The Zwitter ion is when the amino acid has a net neutral charge. And so you see it does have charges on it, but the net charge is neutral. So a plus and a minus make 0. And note that this pH is at 7. Okay, at pH 7 we have our Zwitter ion, and Zwitter ions can be acids or bases. Okay, so here's our Zwitter ion right here, and how do we know that that's our Zwitter ion? It's because our net charge is 0. Note that at a low pH, so pH below 7, we have a positive charge. At a pH of a neutral pH 7, we have our Zutter ion, and then a pH that's higher than 7, we have a negative charge. And we'll explain that in more detail in a couple slides. Alright, so here's a titration curve. So amino acids have their own titration curves, and there's a couple parts that I just want to identify here. So we have pH here on this y-axis, and then you have pKa. And remember, pKa tells you how badly something wants to give up its proton. If the number is really low, then that molecule really wants to give up its proton. If the number is high, it doesn't quite want to give up its proton as much. And then you'll notice there's a couple different types of pKa's. There's pK1 and there's pK2. So pK1 and pK2. pK1 is for your carboxyl hydrogen. pK2 is for your amine hydrogen. Okay, then we have the Zwitter ion, and that's shown right here. And then we have this new term called Pi, and he has a math definition and a concept definition. His math definition is pK1 plus pK2 divided by 2. His concept is that this is the pH when your amino acid is neutral. Okay, so it's the pH when we have our Zutter ion. And we'll note that non-ionizable amino acids have two pKa's, pK1 and pK2. And when I say non-ionizable amino acids, let's just remind ourselves what that means. If we go back to that big chart, remember there's 7 amino acids that have an ionizable R group, a pKr. The other 13, their R groups don't ionize. And so these are all non-ionizable R groups because their R groups will not develop a charge, whereas arginine and histidine, they do have ionizable R groups because their R group will develop a charge. Alright. And then ionizable R groups will have 3 pKs. So they'll have pK1, pK2, and pKr. And so here we go. I just skipped forward a couple slides. And this is an ionizable R group, and so we have 3 pKs, pK1, pKr, and pK2. Okay, this right here, you're going to want to memorize this, and it's going to be really helpful for you to answer certain questions on tests or in homework, asking about, what is the charge on an amino acid, or what is a charge on a certain protein. And so the way I've drawn this out is we have COOH representing that carboxylic group on an amino acid, and then over here it's lost the proton. And then above the arrow I wrote a pH of 2.34, and so this means that below a pH of 2.34 we will have our proton, and above this pH we will have lost our proton. And so here below a pH of 2.34, what do we expect? We expect to have our proton, and that's what's shown here. But what happens if we move above that pH? And so if we move anywhere above a pH of 2.34 right here, if we move up, then we expect to lose our proton, and that's what's shown right here. And then with the amine group, at a pH below 9.6, we will have an H3+, so we'll always have a positive charge. Any pH above 9.6, we will have no charge. And so let's look over here. So at a pH below 9.6, we have that NH3+. At a pH above 9.6, we have a neutral charge on that amine group.